Perfect. Yes, so just to recap there, thank you very much for joining our webinar today on the topic of enabling AI in drug discovery. So we've got a very exciting panel lined up, which is going to get to the essence of building a company in this space, touching upon themes of collaboration, technology, opportunities, and strategy. So if you're not part of the panel, I'd be very grateful if you could turn off your camera and mute while the main discussion is taking place. And please feel free to drop questions into the chat box, or you can wait until towards the end where we will have time to open up to the audience. And just to highlight, I am now recording the session and we will endeavour to make this available as on demand afterwards. So just moving on to show you our panel lineup. So I'm delighted to have Steve Gardner, CEO of Precision Life, who will be our moderator today. And then we will go through to each of the panelists for the introduction. Just to add that Fiona did have some slight technical difficulties at the beginning. So we'll just I'll just check in with Fiona there and see how she's doing. But in the meantime, Steve, I will hand over to you to kick us off. Thank you very much.
Thanks very much, Alicia, and thanks to One Nucleus and SBC for organising this. It's I think it's a very timely discussion, a great topic that is generating a lot of interest amongst potential collaborators, startup organisations, investors, and further out into pharma and healthcare. So I'm delighted to have such a great panel. I'm going to get them to give 2 or 3 minutes of intro to themselves, the company, and maybe position a little bit about how they use AI. But can we go round? I'm going to go round top right clockwise on my screen, which is not going to be very useful to any of you. But Yelena, would you mind kicking us off, please?
Sure. So hi everyone, I'm Elena. I'm the founder and CBO of PharmEnable. My academic background is originally in computational biology and genomics, but I ended up leaving academia and becoming a serial entrepreneur because I wanted to focus on projects that are closer to reaching patients and improving patients' lives. Within PharmEnable, what we're doing is using AI to create the next generation of small molecule drugs. So we use AI to map out the possible chemical space and explore it and find kind of more three-dimensional and complex small molecules that are still drug-like and that can be used to create breakthrough treatments in areas of high clinical need. We're a very collaborative company. We love working in partnerships with other organizations that are working on interesting biology and have challenging targets that we can team up on. Yeah, I'm looking forward to the discussion about the interesting challenges in AI and building up businesses in this area.
Fantastic, thank you, Fiona.
Sure, and I think I've fixed my internet issue, but I hope you can all hear me.
Yeah, we can hear you.
Yeah, so I'm the founder of Repositive. At Repositive, we support biopharma who are doing oncology drug discovery to find the right mouse models and other cancer models to suit their needs. It's especially difficult if you're doing very targeted, targeted stratified patient segments that you're aiming for with your therapies. Then you need to find the right model that fits that particular patient group or that particular tumor type. And that's why the service that Repositive is offering of finding the right exact tumor type across all the different cancer models available across the world is essential to speed up a drug discovery in oncology. From all our interactions with biopharma over the years, yeah, I have perspective on where data is being used to accelerate the discovery, and where there's data, there's all clever ways of utilizing that data, such as using AI and similar techniques. So I'm looking forward to this discussion.
Thank you. Payman's got a little distracted in the background there, but Payman, yes. How about you?
Yes, sorry about that. Hi everyone, my name is Payman Gifani. I'm the founder and CEO of AI Vivo, and we are based in Cambridge Science Park, and we have a cell-oriented drug discovery platform, which means in contrast with the major dominated approach in drug discovery, which is the target-oriented, we have a phenotypic approach. We, instead of looking at a specific target to modulate a disease, which we believe is a very simplistic view of doing drug discovery, because we miss lots of nonlinear interaction among different components of cell biology. We consider the whole cell. We develop a mathematical modeling framework that represents the phenotypic effect of different diseases and also different kind of perturbation. And then how, when we compare with the healthy cellular phenotype, how disease can change this phenotype, how different treatments, including, for example, chemical compounds, can change that cellular behavior changes, and some external perturbation, like, for example, CRISPR. And that representation of cellular behavior, this phenotypic representation, is an input to our prediction engine, which then try to rank and predict some unexpected ways that we can modulate a cell behavior, and telling them from, for example, disease to normal, depending on different disease areas. We have two major approaches in drug discovery. We have our own internal pipeline, and we also partner with other pharma companies. The first approach is a disease-oriented approach, when we start from a disease and try to capture the phenotypic representation of that disease at different stages, turning them to a specific model, which we call it manifold. And the manifold is then used as a input representation for the disease in order to rank candidate compounds that can be used for that disease. And this is the disease-oriented approach. The second one is an asset-oriented approach, which we, on the opposite side, try to rank indications. That's a very short introduction of what we do, but I can cover more during the discussion.
Yeah, well, that's fantastic, and we'll come back to those other approaches in a little bit more detail. But Ajay, last but by no means least, how about your good self?
Sure, thanks, Steve. So yeah, my name's Ajay. I'm the founder and CEO of a company called Implicated. We're based at Stevenage. And what we've done is we've been building a computational modeling platform to model cellular processes dynamically. So we go into a lot of details in terms of modeling cells. And we use this knowledge to discover new biology, as well as identify viable drug targets. So we've initially focused on modeling E. coli. So we've been building a whole cell model of E. coli, and we've used that to identify viable drug targets, and then we started to develop drugs against those. So we've got some early stage pipeline assets in the antibacterial space. And more recently, we've started to look at modeling pathways implicated in cancer. So we're looking at like the MAP kinase pathway and the DNA damage response pathway. Again, what we want to do is model these pathways at high resolution, identify drug targets, then develop drugs against those targets. So as a company, we want to establish a pipeline of early stage innovative drug discovery programs.
Well, that's fantastic. So you can see we've got a pretty good spread of interests in the early stage part of drug discovery. And just for full disclosure, Precision Life does analysis of large-scale patient populations. So we do high-resolution stratification of complex diseases. As Payman said, actually, to find those non-linear additive effects, the combinatorial effects that differentiate one population from another, and we use that in a variety of different ways. So we are practitioners of various AI technologies as well. Anyway, so this is, given the different aspects, I really want to touch on a number of areas, and I was going to go through in a sort of starting at the front end of the traditional pharma process with biology and moving through chemistry and into some of the preclinical work. So I was going to come back to Ajay and start and really ask, in your view, what are the impacts that AI is having on unpicking the complexity of diseases? You know, we're seeing it applied outside of the area of precision oncology and rare disease increasingly. So how do you think about complex disease biology?
Yeah, I guess, thanks, Steve. I guess we're starting off with a fundamental notion that when it comes to biology and understanding what goes on in cells, you know, we're just scratching the surface. There's still a lot of biology to discover and understand, and it's this lack of knowledge that leads to the you know, the drug discovery programs failures, issues that we hear about constantly. You know, the fact that the cost of drug discovery is going up and the failure rate is still high. And as everyone probably knows, you know, the failure rate, the cost is most impactful during the clinical stages. So, you know, there's something like over 50% of drugs fail in phase 2 and phase 3 clinical trials because of efficacy. They simply don't work at that stage, and that's during the expense stage. So what we're saying is that we need to get the drug discovery right at the start of the discovery stage, and having cellular models that can simulate biology accurately at the beginning can help that process. And I suppose an analogy would be engineers who design planes. They simulate planes or modeling planes on computers before they go out and build one. And this is what we should be doing basically when it comes to drug discovery. So that's what we're all about. And I guess the issue in terms of modeling cellular processes has been the way we go about it. It's traditionally modeling cellular processes has been at a very low resolution. It's static. In other words, it's usually concerns statistical association between genes and diseases, yeah, as opposed to being dynamic. So I think what we need to do is get to the dynamic stage of modeling, basically, and we can do that now because of all these fancy life sciences tools that are coming on board now. You know, the genomics, the transcriptomics, proteomics, metabolomics data that we're getting, and that's often high quality data which we can use for the modeling purposes. And yeah, and I guess I mean, a great analogy, and I'm stealing this analogy from a podcast I listened to a few weeks ago, and it's basically, the podcast series is called Bio Eats World, and it's put together by a West Coast VC called A16Z. They use a great analogy where they talk about modeling as mapping, as maps. So where we are with computational modeling of cells at the moment is we're at the paper map stage, which is, you know, which as I said before, is flat. You don't get a lot of information. It's static. And where we want to get to is a dynamic Google map type of approach where you can see things in high resolution. You've got lots of data inputs. You get you get dynamic information. You get information where you can, you know, actually discover things. So that's what we're trying to do with our modeling approach and with the data sets that we can get now.
So yeah, absolutely. And you're kind of touching on things that Payman talked about earlier, both in terms of the new types of data that are available. You know, we have all this transcriptomic, we have really high quality imaging, we have more real-time and single cell data available to us. And I wonder, Payman, you know, just thinking about, you touched on in your introduction on some of the things that you're doing, but what opportunities does AI create to use these new types of data in ways that we haven't applied to discovery in the past?
Okay, yes, that's a very good question and interesting opening part of this discussion, which is we are referring to dynamics. The disease is not one static stage, is a dynamical system that changes over time. Therefore, having a simplistic view of we can understand a disease by reducing the order of this complexity and not considering the nonlinear dynamics is something that we are missing now in drug discovery in general. And the other thing that we just actually mentioned is how we can go beyond correlation and try to find some causal relation between cause and effect in biology. We know that the biological systems are highly complex, lots of concurrent process happening at the same time. We have resource sharing, lots of protein doing and take part in different pathways at the same time. And all of this creating a very complex system that's finding and connecting and inferring a kind of causal relation between these components in order to say, this is the main cause of this observation that we have. And therefore, this kind of nonlinear dynamical system modeling, which we inspired from physics, can be used in order to map all the cellular behavior phenotypically on a landscape, and then try to see the transition and the relation between cellular states and cellular behavior. And then from what you said, the data, the functional genomics data, which is the only kind of data that we use as an input to our engine, is also representing this kind of changes at different levels. Therefore, from the AI side, how we can integrate and represent this kind of various types of data, which sometimes they are not compatible, in a framework that they are ready for any machine learning tools is the key. Therefore, just forget about the AI. How we can have a very clean and representation of a specific stage of a disease is the key, the first step. And then from here, asking different questions, how these connections can be causal, and then how we can have a modeling system that is not just showing them some correlation between these variables. We are entering to the big data medicine, which is actually based on building this kind of knowledge graph, which I believe is just, as you said, it's a static representation of some correlation. That's not exactly telling us what's happening inside the cell. We need to go beyond that. And the kind of tools that you also mentioned, integrating phenotypic approach plus some methods that we can actually use. And again, because the topic is AI in drug discovery, and we believe AI is a buzzword now, which is very confusing sometimes for lots of people. But we believe the systems biology and systems pharmacology and looking at this kind of angle is the key, not just AI. AI is just the tools that we can use. But as you mentioned, there are some companies that try to integrate automation, generating lots of data, representing this dynamics enables us to have input data in order to build that kind of right model that can then have kind of causal inference and hopefully reducing this high failure rate in early discovery or in a later stage.
Yeah, absolutely. I'm certain we'll come back to the question of how much AI is truly enabling and how much it's extensions of existing technologies, yes, how much it's about data collection. I think those are great themes. And in fact, I mean, moving on to Elena, if you look at computational chemistry, I'm old enough to have been in that space in the 80s for quite a while. And it's developed continuously in terms of sophistication over that period. I'm really interested. We do seem to have hit an inflection point where AI is really now starting to drive the business of medicinal chemistry. But what's happened in your mind, and what new opportunities does AI offer for molecular design?
So I think it's a really exciting time to be in a field of AI for drug discovery, because as you mentioned, it's really a time when I feel like AI tools for drug discovery have really come into their own for a number of reasons, and a lot of it has been to do with kind of incremental improvements over time on kind of a variety of different technologies over the last kind of two or three or even 4 decades. So including computational chemistry tools, availability of data that we have, availability of kind of AI tools that we have, computational power. And we're really, at this point, we're at a stage where we can use all of those tools in combination to really fully understand the problem space, and kind of really map out kind of how do we create novel treatments. And I think, we're in an area of small molecules, but I think these technologies are going to kind of impact all types of treatments across all modalities as well. And I think the main promises are around, so firstly, accelerating how we do drug discovery and drug development, and kind of starting with smaller numbers of candidate drugs, and really kind of making the process as efficient as possible, and also reducing attrition rates, as everyone else has already mentioned as well, because if you start off with this kind of rational drug design, you can really kind of start off with an interesting design and well-profiled kind of compounds and potential therapeutics from the very start, and that improves the success rates. And then combining that kind of chemistry AI power with all the kind of fantastic biology research that's also happening, and kind of having the kind of powerful chemistry alongside things like patient stratification, and really understanding the underlying biology of the targets and which populations we can use those treatments for. And I think in combination, that's something that's really going to revolutionize how we create medicines.
Yeah, I totally agree with you. And actually, Neil just raised an interesting question along those lines, which is, we've in computational chemistry, we've come up this resolution pathway since the 80s, where we made really broad assumptions about how molecules worked and how force fields happened. But now we have much more computing power. Is your business enabled by cloud computing and GPU computing and those higher densities? And how much of a solved problem is that? How much further do we need to go with high performance computing?
I don't feel like it's a limitation for us. I feel like we're at a stage where, GPU computing and cloud computing has largely solved these problems of scale, at least on the chemistry end. And it's a case of kind of cost more than I think computing capacity is. It's kind of where it needs to be for solving these problems. So.
Cool. Well, somewhat the other end of the scale in terms of high performance computing is Fiona, and you're really much more involved on the preclinical side of things, and fitting. You've got an exquisite fit that you've got to get between the models that you're using and the patient populations. How do you use the tools to enable you to do that? And how do these models fit into the wider AI strategy that pharma companies have for their clinical development? And you're on mute at the moment, Fiona.
Yeah. So basically, you see that most of the pharma who are applying an AI strategy, they're doing this because they are going for the concept of precision medicine, in the sense that you want to know what you're targeting. You want to know what you're targeting it with. You want to know which pathway you're perturbing. You want to know you have a mechanism of action that you believe is correct, although we've seen in the past that some supposed mechanisms of actions turned out to be something different. But in the sense of precision medicine, it's all about building on your knowledge of the functioning of the cell in order to target very precisely the perturbation you want in the cell. And in case of cancer oncology, you're trying to then basically hit the stop brakes for the cancer, or you know that only cells with a certain fingerprint in terms of their genotype and the expression of different genes will be responsive to your therapy. And as you get more and more precise in terms of your target population of cancers, that means that you're more and more reliant on the data that describes not only the data that you use to arrive at these conclusions, which means that there's much more data generation happening in the early drug discovery process in order to arrive at these conclusions, but also when you then want to select the right model or the right cancer tissue for you to conduct your study, you need to have a high level of confidence that this is actually a tumor tissue that has this particular gene expressed, it has this particular mutation, it has this particular drug response to known therapies. And if it fits all of those criteria, then this is the target for my new therapy. And that's all very good in theory, but if you then can't select a model that has those qualities, then you're stuck, right? Then you've invested a lot in your clever algorithms and data up in the early drug discovery in order to arrive at this very targeted therapy that will be the potential next blockbuster. But if you then can't find the right tissue and the right cancer models to test it on, to actually validate and get approval to move into clinical, well, then you're basically stuck until you find it, or you will have to make shortcuts in the non-scientifically approved way of choosing models that are not actually modeling what you're aiming for. Which then becomes unreliable science. So that's why in the work that we do at Repositive, it's really essential that we know what is the data that is characterizing the different models. Do we have the right genotype? Do we have full genomic information underlying that? Do we have confidence that these mutations are actually present in this model, that they weren't just a fluke by the analysis engine that was used to analyze the data. So it's all about, it all comes down to how much data do you have to support your hypothesis, both in arriving at your target tumor type, but also in then selecting the right models for your study. It's all about, well, how easily can I find the right study based, the right model based on the data available?
I think you're highlighting one of the fundamental, I could call it limitations or enablers of AI, which is this really high quality data, regardless of where it sits on the pipeline, whether, you know, in some of the things that we deal with, whether that's clinical and epidemiological or genomic, whether it's transcriptomic information, whether it's detailed into intersections between chemistry. Those are really interesting and essential parts of the problem, I think.
If you don't mind me adding to that, because it's interesting to see how there's been a shift between the forward-looking biopharma companies would be the ones who were the quickest to utilize what data was available in the public domain, to then moving on to where can we purchase the best possible data to use in our early drug discovery pipeline, to then now you see new startups like Recursion Pharma, for instance, saying, well, how much of the data that we need can we generate in a systematic way using robotics and streamlining our processes to basically generate the data that we need in order to make the predictions that we want to make about the next target for a pipeline. And that's been a big shift that's been happening in the last, I would say 5 years or so.
Yeah, I think that's a really interesting one. It touches on some of the fundamentals of what the market is for these technologies. And I'm going to switch over to some hopefully quick fire questions across the team on these kind of things. I see in our space a lot of companies collecting a lot of data on large-scale patient populations, and they're spending hundreds of millions in doing this. You mentioned recursion, but you could put in vitro, you could put 23andMe in that, the Flatiron, Foundation Medical into those kind of categories. I wonder if, I'm suspecting that nobody on this call has hundreds of millions to throw at that particular problem. So I'm wondering where you think the value can be created. Is it really proprietary data sets all the time? Is it better analytics? Is it, the molecular IP that are being generated, or some models that are coming out of this? Where do you feel that the value is coming from? And I'll go back to Payman quickly on this one.
Yes, that's a very good question. I think one of the major bottleneck that we have is generating more data. That doesn't necessarily mean we have a better understanding of what's happening. And if we are referring to disease as a dynamical system, then generating right kind of data representing different stage of a disease correctly is the key. And what Fiona also mentioned, disease characterization, what disease is actually, which disease model representing one specific aspect of phenotypic observation that we might actually have for a disease. What kind of data is the key? Even the cell lines is the good representation, or animal model is the good one, or patient samples and the data that we have is a source of the information is the extremely important here. And the other things is the data from public domain is diverse. They are generated by multiple different platforms. Sometimes it's very difficult to compare them, very difficult to unify them. And that's another area that sometimes even pharma companies not relying on public domain data because that's not a good source. Sometimes we against that because we already generated lots of interesting insight from the data that we can access from public domain. Therefore, I believe instead of referring to large scale data, generating insightful data that's representing a specific part of the dynamical process for a disease is the key. And as you also mentioned, some other companies like Recursion, they build this kind of high throughput screening system that they use cell lines to generate image data in order to see the difference between and to capture the effect of perturbation, like for example, drugs. This large scale data is useful and definitely something interesting to have a look. But image data has its own limitations because we are just trying to see some aspect of the disease which is not captured. And that's why we have been referring to using functional genomics data, anything after sequencing like transcription data, selection data, epigenetic data, protein level data, which tell us something about this dynamic is the key. But at the same time, generating this a large scale data is expensive. And even if you want to do that, if you look at the disease side, which disease you want to generate that kind of data, if you are looking at the compound side, the compound area is also very big. Therefore, I believe, and or a strategy is how we can use AI to build this in silico model in order to guide this kind of data generation in the next step.
So you're looking at the model as being the value piece. Ajay, how?
As a first step, but the data generation is also extremely important, and we are actively generating data using our specific technology assay.
Mm. And Ajay, are you you're building out models as well? Are you thinking that is where your value is most being created?
Yes, I mean we generate, for example, for our E. coli model, we generate our own data ourselves. So we've got a CRISPR knockout library, a tunable CRISPR that enables us to generate data that we feed into the model. For the for our mammalian work, we're working with a group at Abraham who specialise on a kinase pathway, and we've used data they've generated on a melanoma cell line. So we've used their data alongside data that's publicly available, and I guess what our modelling, what we're trying to do is create a dynamic model. So we're looking at things like the catalytic rates of enzymes, the levels of enzymes that are available, the messenger RNA levels, the half-life of these enzymes, the concentration of these enzymes and messenger RNA. So ours is much more of a dynamic approach, and it's more laborious, but we get better. We feel we'll get more accurate data out of our modelling.
Yeah, no, I totally see that. We've got some questions coming in from the audience, and one of them is a fundamental one that I realise we haven't particularly answered so far. So I'm going to ask for a show of hands amongst the panelists here. But Duncan asked, what's the difference between AI or machine learning and just using computational methods? Well, sorry to disabuse you, Duncan, but they're all just computational methods. We can dress them up and we can make it sound fancier than that, but there are, everything that we do is a clever way of doing maths on problems. But specifically, machine learning is really neural nets, and AI is a raft of other tools. It's a big toolkit, which includes neural nets, deep learning, machine learning, but also includes things like statistical analysis, NLP, natural language processing, or text handling. It includes geometric and logical operations and a whole variety of other things. So, hands up, panelists, who would characterize themselves as AI, machine learning, or just plain old computational methods? AI, who's going to hold out that they're doing AI? That's Payman. I'm holding my hand up as well. Yelena's gone for it. Who's using machine learning? Okay, so Ajay is using machine learning, and Payman as well. Great, we do a little bit. And Fiona, I guess that means just plain old computational methods.
Plain old computational methods and hard work that it takes to scrub the data, which is a big part of doing any kind of data analysis these days.
Huge part of it, absolutely. That's the tough bit. So another question from Kevin is, just on that point, how do different companies access the necessary data to build models? We have millions of academic papers being deposited on a regular basis. How do you deal with this pretty quickly? So Yelena, I mean, you must, for every target that you go after, for every study that you look at, you must have to deal with huge amounts of literature.
Yeah, I mean, we start off with kind of publicly available data and commercially available databases. So all of that is kind of preloaded into our system. We do a lot of kind of custom in-house research for every target that we focus on. I think, I mean, every AI company, that you always want more data. I mean, I don't think there's a point where you kind of wouldn't want to kind of enhance that. But for us, the focus is very much on kind of using the tools to understand the problem space and to really kind of design very precise molecules and solutions. So I think the value is in creating novel IP on our end. But yeah, we're always looking for ways to generate new data, either ourselves or also through partnerships. So we kind of learned from every project that we do as well, and that helps. And if you're an AI company, your AI kind of keeps learning, which is great. So yeah.
Fantastic, thank you. So Carlos asked a big question that I was going to come back to, which is, how does AI, what opportunities does it offer to change the business model of drug discovery organizations? How about Fiona?
I mean, if for the organizations that haven't already moved to a data-driven approach, right? And I call it data-driven rather than AI-driven, because if you have to start with the data, then you can throw the algorithms at it, right? But those organizations that have not moved to a data-driven approach, I wouldn't know where they would fit in any landscape nowadays. I mean, you have to start with a data-driven approach, but then it's just a question of, do you build up a capacity internally on having both the knowledge to use all those magnificent tool sets that are out there to throw at your data, or are you going to rely on service providers who can come in and provide this kind of insights for you on your data? I think is the main question for biopharma companies and pharmaceutical companies, therapeutic companies nowadays. The fact that it's a different type of tool, like that this tool is called AI rather than any computational algorithm, I think is just a sign of the times and doesn't change the core of the question. It's more about, will you invest in having this expertise in-house, or are you going to collaborate or outsource it to service providers?
Well, that leads us into the other side of this question, of course, which is, and I'd ask everybody to reflect on the type of business model that they're using. But as I go round, what's resonating with investors in the mix here? So, maybe Jelena, you're in an industry that's been transformed by these technologies, and it's offering new potential very directly inside a space that traditional pharma would recognize as something that has traditionally been its bread and butter. So how is it changing your world, and what works for investors in this space?
I think it's a very exciting space for investors because there's so many opportunities, and there's a lot of ability to kind of generate novel IP and create value in interesting places through a range of kind of business models people have already mentioned. So kind of generating data, using data in interesting ways for precision studies, generating IP, and also accelerating every step of the process of drug discovery as well. I think there's kind of a culture difference between different groups of investors. So because AI and drug discovery ends up at this kind of intersection of tech and biology, and depending on kind of which side the investors are from, I think they have kind of preferences for slightly different kind of business models and slightly different timelines. So with tech investors, it's a lot more about kind of building up a platform. The timelines tend to be a bit faster, and the amounts of money are different. And then with kind of more drug development kind of biotech investors, they're aware of like the kind of scale of investment that it requires to kind of go down the clinical trials route. So and I think there's a lot of different kind of business models that you can have within that, and the investors you have on board need to be aligned with wherever kind of the ultimate direction of the company is going.
Yeah, and Ajay, how are you thinking about developing your business? In a way that's going to be investor friendly, and what kind of business models are you using to do that?
Sure. We fundamentally believe that the value is in the drug discovery program. So it's all about generating IP around the drug program. So that could be a composition of matter against a novel target. So that's where we see the value. That's the strategy we're taking. And in terms of investors, we're out fundraising at the moment, and as Jelena mentioned, you've got the tech investors who are starting to get interested in what we're doing, as well as the traditional life science investors out there. And from the exposure I've had speaking to investors at the moment, what we're seeing is that tech investors are interested in the platform and how it delivers, and with the life science VCs, that they're still focusing more on where we are with the drug discovery program, how close are we towards the clinic. So they want to see a better data package on the drug discovery program that we're aiming to move towards the clinic. So that's what we're seeing at the moment.
Okay. Payman, I'm going to ask you a slightly different question. I'm trying to get through some of the questions asked by the attendees. So Ruben was asking, reflecting on the fact that cancers mutate and become resistant to drugs, and he was wondering if AI is a solution to hit some of the survival mechanisms. Now, I guess that is really well expressed phenotypically, and the kind of tools that you're using may offer some way of perhaps preventing escape from these kind of mechanisms. Have you looked at any of that?
Yes, exactly. What we are trying to see here, we define, as I explained earlier, we define different stage of a disease, and then we have the perturbation. Or if we consider the cells as a healthy state, then the disease is deviation of this healthy state to have a different kind of behavior. And that kind of behavior then represented at different diseases. And then when we also have some mutation, and that mutation is also a representation of a functional effect in that cell. And when we have treatment, the treatment is also trying to change that behavior. Therefore, if we manage to capture, and that's exactly what we're trying to do, capture this change in cellular behavior, which is actually happening from normal to disease, disease when treated by a drug, and then when we have a resistance phenotype, which again, a specific stage of a disease which doesn't respond to that treatment. All of that, if we capture that transition between these kind of conditions, looking at functional genomics data, then we might actually have some clue that what needs to be modulated now. And that's leading to novel way to trigger that disease, how we can bypass the resistance, how we can, at the same time, which part of the disease we need to modulate in order to improve the efficacy, which part of the disease we need to modulate in order to reduce the toxicity and also resistance. And as a highlight to what we also discussed on the disease side, on the data side, having a right kind of data is the key. And having different data that represent different conditions is also very important. And what we did is creating a huge map of different disease and cell stages, which we currently have something around 800,000 different models that's representing different cellular conditions and the transition between them. And that's somehow helping us to see what is the missing data. And then generating new data is also very key here to have some toolbox to answer this kind of questions.
Yeah, absolutely, absolutely. I've got two questions that I'm going to pick up in one answer, one which are from Miranda: how does AI model the impact of a designed therapy on tissues beyond those that are targeted? And also from Robin, saying, with AI helping to improve the success rate of clinical trials, do you see it ever replacing and shortening clinical trials? Those are two areas that we're very interested in, and they look at the impact of disease biology and how it expresses across a patient population. So Payman earlier mentioned efficacy, and Ajay mentioned it as well. Being able to understand the influences on disease biology and how homogeneous or heterogeneous they are across different patient subgroups is a real insight into how likely you are to get a single signal coming out in a clinical trial. If you can define patient stratification biomarkers that allow you to identify those patient subgroups really accurately, to Fiona's point earlier, then it gives you a much better chance, I think, of being able to understand how to design a smaller clinical trial that's easier to recruit, is faster to respond, and actually generates more meaningful signal, cleaner signal. So that was me doing a very quick answer. There is one question from Elizabeth, which I was hoping we were going to get, which is probably a good place to finish up here, which is, how do we find people to enable us to run our businesses and be competitive with those lovely US behemoths that we love to compete against? So how do you go about getting good people? And I do need 30 seconds from each of you on this one. Fiona, how about yourself?
would say that AI and if you just say the words data science is so popular that there's lots of people who are looking to get into this field. So you have lots of opportunities to hire young talent that, as long as you have someone in the organization who is more experienced and needs extra hands from time to time to do something, you can actually train your own talent in the skills that you need in these areas. So those are my 30 seconds.
Thank you. And Yelena, are you pulling them all from the same background or different backgrounds?
I think it's different for us. So we have a kind of virtual biotech remote-based model. So for us, that's been really enabling because it means we can hire talent from all over the world. It doesn't matter too much where they're based within a certain set of time zones, just for kind of practical reasons. And we have quite a specialized team. So it's computational chemists, medicinal chemists, and biologists. But we've been able to hire fantastic people because of how flexible we are in terms of kind of where the people are and how we work.
Thank you. Ajay?
Yeah, I mean, we've managed to tap into early stage graduates really. So because our founder is an academic, he usually spots these good graduates and tries to incite them into our companies. So that's the route we've taken to date.
Payman, have you managed to find the people you're looking for?
I think sometimes the challenge for us is how we can find someone with background in biology and also computational side and AI. Sometimes having one person who actually have all of this is the key. And we were lucky to have great people with systems biology background, and which they can connect and then they can communicate with both the disease experts, the biology side, and the chemical side. Therefore, I think we're actively looking for people that they have this kind of multidisciplinary background, because they can just communicate and they can bring some ideas from one field to another field.
Well, they're the unicorn. Yeah, they're the unicorns, exactly. Of course. Yeah, and we're all after them.
Yes, definitely. Yes.
I thought you might be naming names. Anyway, look, we could talk for ages about this stuff. We've really only scratched the surface. I hope we've done a decent job of airing some of the issues. But I want to thank all of the panelists. I really appreciate your input into this. It's been fascinating to hear all of these different perspectives on the problem. And hopefully I'm going to leave 4 minutes instead of 5 for Hugo to finish off with some information from the and some of the new initiatives that you're kicking off.
Yeah, super. Thanks, Steve. Well, I mean, first of all, I'd like to thank all the panelists, Yelena, Fiona, Ajay, and Payman, and our moderator, Steve, for the day. And I'd also like to thank Alicia for organizing this One Nucleus event with us. So I think it's been a really useful panel talk, and I hope you'll carry some of these conversations offline. So I'm just going to try and share my screen, see if that works. Yeah, so I mean, to those who don't know, the Stevenage Bioscience Catalyst is a science park located in Stevenage on the same grounds as GSK, and we're one of the leading locations to develop cutting-edge therapeutics, and we're home to the Cell and Gene Therapy Catapult and LifeArt. And since the inception of SBC in 2012, our occupier companies have received about 1.6 billion in equity funding, grants, and acquisition, and so we have a wide variety of startups from pre-seed all the way to IPO now. So I'd like to talk a little bit about the project that we have that's really relevant to the topic at hand. Is that we're starting an accelerator program aimed at the intersection of data analytics and advanced therapeutics. And we've aptly named it Data-Driven Advanced Therapeutics Accelerator, or DATA for short. And this will be open for any startups that are in the field of drug discovery, genomics, or advanced therapies and applying AI or data analytics. So going to the next page, the program is going to be run for 20 weeks, and it's going to be a virtual program because of the pandemic and the current circumstances at this point. And the startups will have a chance to learn and benefit from the SBC community, who are quite experienced in commercializing therapeutics. So during the accelerator program, there will be a lot of panel talks such as this one, and followed by some networking opportunities with panelists for people who take part in, for startups who take part in this program. And we'll have sessions focused on pharma, the different established business models, as well as the emerging ones in the field of AI-enabled drug discovery. And most of the sessions are going to be quite specialist, aimed for startups within the field, and cover topics such as approaching pharma, getting a JV, talking to tech and life science investors and knowing the difference, and also cover more traditional sessions, I suppose, as, on IP, pitching, and as well as culminating in a demo day at the end of the program. So who's it for? It's for startups that are pre-seed, well, pre-seed to seed, so pre-series A, but you must have some sort of MVP or proof of concept. And the focus is data-enabled drug discovery, genomics, advanced therapies, and your startup. And we're looking for a minimum of 2 co-founders, because in our experience, that's generally a single founders. It's a bit tough to attend all the talks and get the most out of the program. And the application will be open in about 2 weeks, on the 15th of February. And the launch event will be on the 10th of the May, so look out for that. And for the time being, our website will be live on the 15th of Feb. So for the time being, if you need more information on this, or you'd like to get involved, I don't know, as a mentor or even as a panelist, or just get more information to apply as a startup, you can contact me or go to the Stevenage Catalyst website on the Contact Us page. And I'll leave that over to Alicia now, I think. Oops, if that's possible. But thanks for everything. Yeah.
Thank you very much. Yeah, it's just left to me just to say thank you to the panelists, to Steve for moderating, SBC for your support. Incredibly useful discussion. Definitely room to continue into and explore some of the themes that were brought up into more depth, and also to the attendees for lots of engaging questions. So it was a wonderful two-way conversation, which is perhaps one of the best ways to get the most out of these sorts of events. So thank you very much for all who have joined, again to the panel for sharing your expertise. We couldn't have done it without you. I do wish you a good rest of the day, and I hope to see you on future webinars. So thank you very much. Take care.
Thank you.